Okay, welcome back to Psych 319, Chapter 2, Part 4. So I'm going to go really quickly through this last bit. We talked a lot about histology and neuroanatomy. Now I want to talk about how we trace neural connections and um, get some activity as well. So there are two ways to find out what parts of the brain are connected to other parts. And you can do that with anterograde labeling or retrograde labeling, which means basically you dump a chemical in one part of the brain, in one part of the brain, and you see what that part connects to. And you can do this in a forward-moving fashion or a backward-moving fashion. Um, so let's say you have this area here, VMH. What is it connected to? You can use retrograde labeling to find out what connects to it, or anterograde labeling to find out what it projects to. And you just inject a chemical into area 1, and it's carried to other parts of the brain by following the length of the axon in a process called axoplasmic flow, which is normally used to move nutrients up and down neuronal axons, but you can also use it to transport this tracer. I'm going to skip this stuff. We can't always do some of these techniques that we've talked about in the human brain. You don't want to go in with electrodes and chemicals and things like that. So we do what's called neuroimaging. And in neuroimaging, there's many different types of techniques. You've probably heard a lot in medical shows about CAT scans. A CAT scan is really a CT scan, a computerized tomography. And it's really like a three-dimensional X-ray. So you get a bunch of X-rays of the brain, and you reconstruct them to create a 3D model of whatever you're looking at. So here are the two-dimensional slices of the image, and you can use that for perhaps detect a mass in the brain by differences in density. This is the inside of the machine, so this revolves around the axis and can take pictures in many orientations. So let's skip to the chase here. And then you can get this 3D reconstruction. So this is what you'd get from a traditional X-ray, or if you just took one individual slice of the CT. But if you combine it in 3D, you get a very elaborate image of the structure you're trying to look at. But it's mostly good for things like tissue and bone. We don't get things like neural activity from CT. An MRI is a very great technique for the brain. Because unlike X-rays and CT, it doesn't use any radiation whatsoever. It's, to our knowledge, after 50, 60, 70 years of doing this, there are no known side effects for using MRI. It's a very complicated technique, so I won't describe it in detail because it goes beyond the scope of this class. But just know it's safe to use. It doesn't use radiation. I have some hints about how it works in the slides, but I'm not going to talk about that now. But you can get either a 3D or 2D image that gives you very rich anatomy of the human brain. Another thing that we can do that's not invasive is using psychophysics. So psychophysics is just a formal name for measuring behavior. Psycho meaning in the mind and physics meaning in the outside world. And we want to measure behavior because we want to characterize behavior in a very accurate way before we go in and start creating physiology or in getting measurements of physiology. So you usually do this as the first step before conducting anatomy and physiology. There are many ways of doing this, which I'm just going to hint at here. For example, the detection threshold method would show a bunch of dimly lit lights, and some of them you'd be able to see, and some of them you wouldn't. And when you find the point at which a person just starts to be able to detect the light, then you can say, that's their detection threshold. We know exactly the point at which they just start to see dim light. But anyway, it's about measuring behavior. And so imagine you had a dimly lit light and you were increasing the intensity from darkness at 0 to 45 candelas, we'll say. Those are the light units. At some point, so down here the subject says whether they see it or not. When it's completely dark, they say, well, I don't see it any of the time. And as if it's very bright, then they can detect the spot 100% of the time. Well, somewhere in between on this curve, they can see things about half of the time. So right around 24 candelas, the subject might say, sometimes they see it and sometimes they don't. And so we say this is their detection threshold. that it's the point at which people can see or detect things about 50% of the time. So back into the physiology. Um, in rats, you can record neural activity by dropping an electrode into the brain, either in a one-shot deal, or you can do that by embedding the electrode in a connecting, in dental acrylic with a connecting socket, and you can record on a daily basis from the same location. This is called electrophysiology, otherwise known as extracellular recording, because you're dropping the electrode near neurons, but not exactly in the neuron. Intracellular recording is a lot more delicate, and you usually do that in a culture, in a slice of tissue, where a glass pipette is actually affixed to the neuron itself or inserted in the neuron. So, skipping ahead. So this is intracellular recording, otherwise known as the patch clamp technique. You have a glass pipette, and it's placed right next to, I'm getting this whirly button here. I wonder what button I pressed. You get this glass pipette, and it's pressed right next to the cell wall, and it forms a little vacuum that allows you to record the electrical potentials inside that. And then those electrical potentials show up as a waveform on an oscilloscope, and you can isolate a single waveform, and then you know you're recording from a single neuron. It's pretty magical stuff. So normally we record multicellular recording, which is recording from many cells at once, but single unit recording is what I just described there, where you can look at one neuron at a time. Okay, so in the human brain, again, we can't do that, so we tend to use something called EEG, electroencephalography, and that is, the quintessential psychology experiment where you have somebody who's thinking about something and have all these silly electrodes glued to their scalp. Well, they're not so silly. They can actually detect neuronal activity through the skull. Now you can't tell exactly where things are coming from. I won't be able to tell you what part of the brain is responsible for a particular thought, because the spatial resolution of this technique is actually not very good. However, it's really great at telling you when something occurs. It has great temporal resolution. So it's oftentimes used in sleep studies, where we're mostly concerned about when somebody's overall state of attention is changing over time. You can visualize, so here's another example about a fancier version is magnetoencephalography, MEG. A similar idea. The technique is very similar. Okay, so let's get to recording activity. A lot of what we've been talking about has been anatomy up to this point, except for EEG and MEG. In a classic experiment, what you would do in a rat is you take a slice of a rat brain. Well, you would inject a radioactive tracer into the rat, and that radioactive tracer is bonded with a sugar, and the neurons will, when they need fuel, they drink up that sugar, they use it for fuel, and then they're subsequently labeled with the radioactive tracer. Then you can slice the brain and process it like you would a photograph, and you'll get this really cool image that shows you which parts of the brain were active during that task, like in this little region down there. This is called autoradiography. 2-deoxyglucose autoradiography, very complicated name, but it's essentially a labeling technique, and it can tell you when cells become active. You can do this in humans using a technique called positron emission tomography, or a PET scan. So you've probably heard of this through television and in some of your other classes. A PET scan will measure neural activity, and it does use a radioactive tracer. It uses usually a radioactive gas or an injection, although it's very mild and a rapidly decaying radioactive substance. So usually the activity or the radioactiviness of it decays in 45 minutes or an hour or so. And so you can detect activity using a similar type of paradigm. Here a subject is just clenching their fist, and you can see which parts of the brain light up when they're doing that. But same logic as autoradiography. Now you can also detect activity using MRI, and this is really the technique that we currently use the most. MRI, again, is non-invasive. You don't need radioactivity. You don't need to ingest any chemicals or radioactive substances, and you can scan somebody again and again. And there are no known side effects with this technique. And you can project, you can find these regions of activity and put them atop anatomical regions, so you can start to associate which parts of the brain light up during a particular task. So traditional MRI is a static snapshot of the brain, but MRI is a dynamic movie of the brain's activity. And a technique that we commonly use is called the blood oxygen level dependent technique, the BOLD technique. And it measures the difference between blood that's been used and blood that hasn't been used. They have slightly different magnetic properties, and the machine can actually detect that. It's pretty cool. Now, we can't do lesions in humans, but what we can do is we can simulate that by using a giant magnet. Transcranial magnetic stimulation, it creates a magnetic pulse that temporarily disrupts electrical activity in the brain. It's not so much that it would cause a seizure or any damage, but it's just enough to confuse neurons in a temporary state. Now, it's not very specific. You can't knock out one or two neurons. You're going to be knocking out huge sections of the brain. So it's not incredibly useful, but you can learn a lot of interesting things from that technique. But it does have unreliable localization. As I say, it's about as elegant as cutting a wedding cake with a baseball bat. I mean, you're going to learn something from it. It's going to get the job done, but it's not going to be very pretty. And then the last technique I want to talk about are targeted mutations. You can take animals and breed them to lack specific genes that would be used to normally produce a protein or an enzyme in a cell. And as a result, these genetically modified mice or rats are deficient in a particular behavior or physiological function. Then you can measure the effect of that behavior. And a lot of science is really heading in this direction, in biological science particularly. What we want to see is what genes are responsible for diseases that causes changes in behavior. So the target may be located at one part of the brain that's critical for function, like in the hippocampus or memory, or it could be a global thing that affects all areas of the brain, something that might affect the production of a neurotransmitter, for example. So we call these knockout mice because they have genes knocked out of their sequences. And you might consider this some kind of genetic lesion. And again, the hope is that we will find genes responsible for major diseases like Down syndrome and so on and so forth. So these are two mice, I'm not sure if they're Zucker rats or not. These might be a different strain, but essentially they only differ by one gene, and that gene seems to account for obesity in this species. If the gene is present, the rats will overeat and they get rather hefty size, and if that gene is not present, then the behavior is totally different. It results in a dramatically different quality of life for the animal. So just one example, but there's most of the major diseases that we're looking at are using this technique: Alzheimer's, Parkinson's, autism, and so on and so forth. We're trying to find the genetic correlates of the physiological underpinnings of the behavior. Anyway, thanks for your patience, and I hope you learned something from these slides, and I will see you again in Chapter 3.